Yes, we're going to introduce INVAM. So INVAM is the largest culture collection of arbuscular mycorrhizal fungi in the world. Terra, Peggy, Liz, and I are curating it. It's here now. It's been here for what, 3 years? And so I am going to introduce the value of mycorrhizal biodiversity, and then Liz is going to show examples of the application of mycorrhizae, and then Terra is going to show what we do to maintain INVAM. That's our order. But interrupt us at whenever you want. It's all good. But for those of you who don't know, this is an arbuscule. This is the clear root. This blue cloudy thing. It's actually a branching structure called arbuscule, Greek for bush. Okay, so you all know a lot about mycorrhizae. I'm just going to do a really super quick introduction. They have hyphae that branch out from the root where the arbuscule is, and site of exchange of resources from the fungus for the plant to the plant. So carbon comes from the plant to the fungus. Fungus is feeding phosphorus, nitrogen to the plant. And because of this, plants get bigger. These are pictures from Scott Mangan from his dissertation work. Big plant, little plant, big plant, little plant. Big plant is with mycorrhizae, little plant is without mycorrhizae. Very common result. And mycorrhizal fungi can also enhance host defenses. They can alleviate drought stress. They can limit uptake of heavy metals. They can aggregate soil. This has consequences for populations, altering the composition, altering the compatibility of plants, consequences for community, altering the dynamics of community interactions and trophic levels, has consequences for ecosystems, enhancing soil stability. It's a major driver of cycling in these systems. And because it can influence soil carbon storage, it can have global consequences. And so the way ecologists have typically thought about mycorrhizae is just all around good guys, and sort of like vanilla ice cream on pie. So you get pumpkin pie or apple pie, and you get a la mode. You just get an ice cream on top, and it makes the pie better. And that's sort of the spirit of a lot of the work of mycorrhizae in ecology. And so what we have done, and what INVAM is all about, is the different flavors of ice cream. So different species of mycorrhizae have different effects, have different consequences. So here's a simple illustration of that. So this is a result of Alison Bennett's dissertation work. So here's sterile spores, and this is defensive compounds in ambient and then exposed to herbivory. So this is induced response to herbivory. This is their aeroid glycosides in Plantago. And then these Plantago are associated with one species of fungus, all from the same field. It suppresses the induction of defense. This is exposed to another fungus. It elevates the constitutive and the induced defense. This is another species of mycorrhizal fungi. This has constitutively elevated defense. You put it all together, you get something, this is a mixture, something like this. So 3 different species of fungi, 3 completely different defensive phenotypes of the same plant. So that's the general spirit of a lot of the work that we've done, which shows that AM fungal species differ in their overall effectiveness in plant growth, their host specificity of that plant growth promotion, their effect on host defense, ability to confer tolerance to drought or heavy metals, and their effect on soil aggregate stability. And because of that, the composition of mycorrhizal fungi will impact ecology. Mycorrhizal fungi also differ in their host specificity of fungal growth rates, their compatibility for roots, their sensitivity to disturbance, soil factors, seasonality, and climate. And because of that, the mycorrhizal fungal composition will be dynamic over time. And the challenge that we all have to understand how this important interaction will function is to understand how these two things feedback, how the change in composition will alter the function of these things over time. And that's a major challenge. To do that, you have to be able to culture, isolate, and maintain cultures of these fungi. That there's, to test any of the different impacts of these fungi on all of the things we care about, we have to be able to isolate and test them. And so these are spores of asexually produced fungi. This is 9 different species, all isolated from the same field. This is where Peggy and I did our dissertation work. So the problem is isolating these fungi is a massive effort, and maintaining them is also a massive effort. So few scientists have the skill set to enable them to do that. And so to facilitate the research on this, we have to have a bank of cultures. Just to illustrate how the status of the global discipline, our lab's collection of AM fungi is, I think, the 3rd largest collection of AM fungi in the world. INVAM is the largest collection. So that sort of sets up the value of the culture collection, is that we need to have these culture collections to learn not just the ecological, but the genetics, everything about biology of these fungi. And so now I'm going to quickly illustrate the value of biodiversity with some of our work, and then Liz is going to take it from there. So a lot of our work is focused on the tallgrass prairie, which you all are familiar with. You know that tallgrass prairie is succeeded by corn because it's very fertile soil. If you abandon corn, it's succeeded by weeds. If you throw the seeds of this prairie over into this post-agricultural soil, you get a dominance of grasses. This is a restoration at Fermi National Accelerator Lab. And so this is typical of most restorations, that they fail to reproduce the original diversity. And in particular, plants vary in life history. These weedy plants that allocate high to reproduction, fast growth, they have no problem establishing in these restoration contexts. What's missing are these very slow growing late successional plants. Tallgrass prairie happens to be super rich in this type of life history, but we have a hard time getting a lot of these species established in these restorations. And so we've asked the extent to which interactions with the soil microbes, in particular mycorrhizal fungi, can explain the limited success of these late successional species. And so doing experiments where we take plants that range from annuals to long-lived perennials, and then inoculating them with the cultures in fungi that we've derived from prairies. And this is Liz's dissertation work here, showing that early successional species are not responsive. This is the log rate. This is the ratio of inoculated, the mass of inoculated over uninoculated. So when the ratio is 1, or there's no effect of inoculation. So late successional species can be very responsive. Each of these dots are the average for an individual species in these categories, and one of the dots is off scale, it's 40 times bigger. So that was 37 species, I think, and now Liz has collected data on 200 prairie species of the Midwest, and non-natives act like early successional native species, and they're not responsive. But this is the biodiversity part. That was all the response to the vanilla ice cream. If you look at different flavors of the ice cream, these are the mid or late successional species here. They're arranged according to the average response. These are the 6 early successional species. In this experiment, they did not benefit from mycorrhizal fungi. But what I want you to direct your attention to is the variance between the different individual isolates of mycorrhizal fungi. Early successional species, not much variance. Late successional species, a lot of variance. Variation is expected to increase with magnitude. So if we adjust the variance by the average, you get this coefficient of variation, and it's still much greater for late successional species than early successional species. That's with 12 species. If we get all the data that we can put non-natives, early successional natives, late successional, late successional species are much more sensitive to AM fungal identity than non-natives or early successional species. So with late successional plants being missing from restorations, they're more responsive to AM fungi, they're more sensitive to AM fungi. Do they contribute to the limited success in restorations? Liz is going to tell you that. I'm going to just tell you that we also find that late succession plants are better hosts for mycorrhizal fungi and better hosts for good mycorrhizal fungi. And so that generates a positive feedback with succession is the qualitative dynamic. And that qualitative dynamic means that that the mycorrhizal fungal dynamics can accelerate succession if you get it right, or it can inhibit succession if it generates alternative stable states in the succession process. And anyway, and then Liz is going to show you the inoculation studies.
Hi, everyone. I'm Liz, for those of you who I haven't met before, and I'm an assistant research professor at KBS. I'm hosted by Jim's lab, and I'm also the owner of a small business called Mica Bloom, where I sell mycorrhizal fungi, so I have a lot of interest in them in many dimensions. So Jim kind of went over this a little bit in talking about mycorrhizal communities across different land use histories. And so in the heart of my research studies mycorrhizae in these two environments, in prairies and in lands that were prairies and are now no longer prairies, which are typically post-agricultural soils. And so what we know about these spaces, things we do in agriculture can really harm these mycorrhizal fungi. For example, tillage can chop up those hyphal networks and kill the fungi. Planting of certain crops, and even crops that are non-mycorrhizal, can ultimately result in communities that are less abundant, less diverse, and comprised of more rural species. And so this is a problem when we're thinking about changing the shape of these spaces from agricultural into something else, whether it be another kind of agriculture or a restoration, for example, because most of them occur, on top of this disturbed soil community. So what I do and what I've done since I met Jim and been in his lab for 14 years for the most part, I collect mycorrhizal fungi from remnant prairies, bulk propagate them in the lab and apply them out in different contexts. So for example, for prairie restoration, rather than transporting just the seeds, I'm transporting microbes also to this environment and then looking to see what happens. And we call this movement of the microbes a microbial transplant. And the stuff we move, the mycorrhizae or any of the microbes, inocula. And so this is a really outdated map of just some of the restoration experiments that have used this technique in the Beaver lab. There's been many, many more, especially in Kansas since we moved here a few years ago. And for anyone who knows me, if it's a plant, I will inoculate it. So I have a ton of data. And I'm just gonna present on a few studies that I've done in Kansas since I moved here in 2016. So first, before I jumped into the field data, I wanted to just demonstrate, this is an example of some perennial agricultural crops, looking at the mycorrhizal response in the greenhouse down here in the field, and they're pretty strongly correlated. So Jim presented a lot of greenhouse data on mycorrhizal responsiveness, and this just shows that, at least for some plants, what happens in the greenhouse can correlate to what happens in the field, because these plants really require their native prairie fungi specifically. So the first case study I want to show is looking at what kinds of inocula we could include in a restoration for transplant. And for this case study, we went to remnant prairies and collected whole prairie soil. So that would have all the stuff that's in soil, mycorrhizae, rhizobia, nematodes. There's a nice worm in that handful of soil that person is holding. So whole prairie soil. I also isolated just the fungi from that same soil, and then we had plots with no inoculation. And we moved our inoculants on the roots of little seedlings. So this little plug, it's a little pot, and we inoculated about 10% by volume with our living microbes or no microbes. And so we replicated this experiment at 5 sites across the Prairie Range, including 2 sites in Kansas. And we monitored 3,200 plugs in total, which were all late successional plants. And then we seeded all these sites with the seed mix native to that area. So let's look at what happened to those inoculated seedlings first. This is some data from the first year. On the top is the proportion survival, and on the bottom is plant growth. And so as you can see, plugs that had AM fungi in black or whole soil in gray grew better and had better survival with that living inocula than the non-inoculated plugs. We transitioned to look at what happened with that recruited seed mixture. We assessed this about a half meter away from those inoculated plugs. And on top is plant community diversity. And on the bottom is the proportion of that sampled plot that was reinvaded by the non-native plant species that was dominant at that site. And what we're showing here is that inoculating with our AIM fungi both improved diversity and reduced non-native proportion in the plots, which is really cool. So I think this data set, it's really powerful because it spans the prairie gradient. We used local inoculants from across that gradient, and it was very consistent across site, and it shows that native prairie fungi might be the most important microbe to include in restoration, specifically for these late successional plants. Which is important when we think about the prairie. It's really highly disturbed. This is a map of just Illinois, and it used to be before the advent of the steel plow, 60% covered in prairie, and now is 0.01%. And so if we think about using microbes as a tool in restoration, whole soil is definitely not an option for a space that has such limited donor soil available. But this data set showed that culturing native prairie fungi is a viable tool. So now that we have decided AM fungi are the most important microbe to include in restoration, the next question is, how much do you have to put out there? And I like to call this my handfuls to dump truckload study. And so what I did, it was an old ag field at the perennial field station that the Land Institute owns here in town. So it's an old brome field. And I took that site and I seeded it with a diverse prairie species mix. And then I established plots that had anywhere from no mycorrhizae to handfuls of mycorrhizae, 8 or 30 kilograms per hectare, all the way to 8,000, equivalent of a dump truckload of this inoculus spread in these plots. You could literally see it on the soil surface. It was very thick. And so I've been monitoring these plots yearly for about 7 years, and so has Reb. Reb's done some work in these plots. And they're really beautiful. I just wanted to show you what they look like year 7. They're really forb heavy. I did about a 10-to-2 forb to grass mix, so they're very showy plots. And I'm going to be showing you quite a few figures that have this same scale at the bottom, which is my inoculation density gradient, having no prairie fungi, handfuls of prairie fungi, all the way to dump truckloads of prairie fungi added into these plots. And so this is a bunch of ways to look at diversity. Pick your favorite. I have FQI and total CC on top, and inverse Simpsons and Shannon on the bottom. They all show the same thing. The more native prairie fungi you add at year 7, the higher diversity your plots are, the better quality that they are. When I dove a little bit deeper into this data set to see what plants were driving this effect, here is something interesting I found. The 2 panels on the left, the 2 graphs on the left are non-native plant abundance and richness. And with increasing densities of native prairie fungi, non-native abundance and richness declined. And the opposite was true for late successional plants. So we saw increasing proportions of these plants in the plots. And there was really no effect for early successional plants. They did pretty well, regardless of what kind of microbes were there. And this pattern was driven by the establishment of late successional legumes. So many people think about legumes, beans, and think about rhizobia being the most important soil microbe for these plants, but this data set shows that prairie fungi are also really important for these plants. So I'm going to shift gears a little bit and talk about some of my later work. I had an NSF entrepreneurial grant supporting my small business, and doing some R&D to see if these prairie fungi have any applications in agronomic settings. And so for that, I worked with organic farmers around town, asking how much do crops like our prairie fungi. And so the first data set I want to show you is some that I collected with a farmer, Scotty Thelman, who was just here a few weeks ago, Juniper Hill Farms. You can see his farm on the way to the field station. And the first experiment I want to show is in-earth assessment of peppers and tomatoes. So this was conducted in a hoop house. Those of you that don't know what a hoop house is, it's a semi-permanent structure that's almost like a greenhouse. So it has a roof, which helps heat it up and, protect its sun from wind and rain. It has irrigation, which runs along the rows, and a weed mat to block down the weeds. So unlike a greenhouse, there's an earthen floor underneath this weed mat. So there are fungi at the site, but they're the disturbed fungi that are typical for agronomic areas. And so I had all these varieties of crops, and down this row, I established blocks of inoculated and non-inoculated plots. So every week, I would go through and collect all the peppers and tomatoes from these plots for every block and weigh them out, their wet biomass, and then hand them off to Farmer Scotty, and him and his team would sell them at the farmer's market. So there's a good chance that you ate some of my research if you shop at the farmer's market. And I'm not gonna jump into the details for every tomato and pepper, but just in general, this is what I found. Inoculated plants were in black and non-inoculated are in gray. If you had inoculation with our prairie fungi, we did see an improvement in crop biomass, which was surprising. I was really excited about this data because most crops are thought to be way less sensitive to mycorrhizae than, for example, our prairie plants. So I was not expecting this at all, and it was really cool. So I dove a little bit deeper, thinking about not just the effects of having prairie fungi, but how important are specific species of these prairie fungi. I just pulled out one piece looking at chard. This figure here, the little blue bars are individual species of fungi. Black is the mixture and white is the non-inoculated control. So this demonstrates that the presence of just even one particular species of fungus has a really profound effect on the biomass of these plants. And these things were correlated with a bunch of traits that I looked at these plants, not traits, that's not the right word, characteristics, because I assessed macro and micronutrients in the leaves of this plant. So for example, plants that grew really big also had a lot of carbon and nitrogen in their leaves and lower potassium, for example. I also partnered with another farmer in town his company is called Valley Cannabis to look at the effects of our prairie mycorrhizae on cannabis. So this farmer grew crops for CBG. And so that's this panel at the bottom left. Same sort of setup, the blue are individual fungal species, black is the mix, white is the non-inoculated control. And we found that individual species really affected how much CBG could be extracted from these medicinal cannabis plants. And I'm glad you came, Kelly, because I put this figure up there for you, thinking about echinacea. Kelly had to connect with some people to extract caffeic acid from echinacea, which is a medicinal compound. And so we saw similar things for cannabis and echinacea, where individual aim fungal species really affected the concentration of these medicinal compounds. So all this is really exciting, especially this stuff with the crops. I was not expecting these findings to be so awesome, given that most crops are less sensitive to mycorrhizae, but obviously they do really affect them. So because of that, there is this massive and growing market for microbial inoculants across the world. Here we are in 2024, and there's 10,000 million, which is a really weird way to say $10 billion is the state of the global market today. Thinking about US, it's a huge chunk of this market, probably around 4 billion. And there's products out there that target a bunch of different crops that we eat and use for agronomic purposes. So I want to show you some unpublished data that is really, really, really fresh, that I'm super excited about, where I assessed some of these commercial inoculants. And so here I'm showing you colonization of 13 commercial inoculants. Their little bars go from, you can't see them because there was no colonization of these fungi. Down here, so those are all my commercial products. I also included a field soil and then our lab-grown prairie fungi. So as you can see, I guess you can't read my words there, but these commercial products often have either extremely low or no viability, or for some reason are dormant. For whatever reason, they are not infecting our plants, in contrast to our lab cultures. And so I also assessed, are they beneficial or not? So another thing about these inoculants is many of them have abiotic things in them. Some of them are good, like fertilizers, and some of them are strange, like vitamin E at a really high level for whatever reason. And so this is looking at crop response to those inoculants. And you can see for the commercial product, there's this huge range of effects. Some are super negative, with the stars are significant. So some inhibit crop growth. There was one that helped. And most of our prairie fungi either were benign or helped crop growth. So similar to some of the data I've already shown. So this study is similar to many that have been out there. You don't have to read them, although one was by our very own Eric Duell at the bottom, looking at these commercial inoculants because they all find the same thing for the most part. These products are dead across the globe. I wanted to point out one at the bottom. Walter asked the question, do arbuscular mycorrhizal inoculants contain the species that they claim? And the short answer was nope. So that was pretty exciting. So not only are they viable, but if they have fungi, it's not necessarily what was listed. So yeah, that's all really scary. So I showed you some evidence in the agronomic world. I wanted to show you one example of commercial versus native inoculants in the prairie. And so this is the data set that Peggy and Sheena started before I got here to Kansas, but I helped them finish it up. In partnership with Monarch Watch, I think Chip Taylor at the time helped advise on selection of some of these plant species and provided them. So we grew a bunch of different milkweeds with our prairie fungi versus a commercial inoculant that they had been using to propagate their milkweeds at that time at AES or whatever it's called now. And so on the left, we're looking at the size of plant, the above ground biomass, with our prairie fungi in black versus the commercial fungi in gray. There was a total shift in plant response. So they liked the prairie fungi and hated that commercial inoculant. We also assessed the latex in these plants by using a capillary tube that essentially sucked out the latex when we punched a little hole in the leaf. And commercial inoculants didn't affect the plants, but there was an increase in latex production for these plants. So anyone who knows the monarch story knows about milkweeds and monarchs and that interaction. This shows that a tritrophic interaction is really important for these plants, and what fungi that these plants are associating with can have cascading effects to the insects that associate with those plants. And this is not unique. There's been a ton of studies out there looking at inoculants and restoration, and they find the same thing. Inoculation source really matters, and native fungi are more beneficial than commercial products. That's probably because most of them are dead based on a lot of that past research. That being said, so just to summarize, my field application studies, fungi can be super beneficial in the field. I showed you across the ecosystems that they can benefit prairie restoration, success, different land management goals, such as reducing weeds. They can also affect our ability to extract medicinal compounds from economically important plants, and of course, affect crop growth. And even though there's all this great evidence from, I focus on my work here, but there's been, a million studies testing these things. The fungal biodiversity is really important, and fungal source seems to really matter. And so I think we have a lot of work to do to help this growing microbial market be more effective. And with that, I'd like to shift it over to Terra, who will talk a little bit about commercial industry when she goes over what we do here at INVAM.
So yeah, Jim and Liz have given the great overview of how important these organisms are to natural ecosystems, agroecosystems, and to application in agriculture and restoration. So that brings us to INVAM. And this is our website. And I wanted to start with our mission statement, which is to acquire, propagate, characterize, and maintain germplasm of arbuscular mycorrhizal fungi in living cultures for preservation and distribution to any person or institution. But also, since INVAM moved to KU from West Virginia in 2021, we've added to provide education through communication and outreach, provide support to researchers by sharing methods and data, and participation in research to our mission. And Jim mentioned that we are the largest collection of arbuscular mycorrhizal fungi in the world. So I wanted to talk about the scope. This phylogeny is from Redeker et al. I think it was published in 2013. And it's on our website also. And so this is the sort of about the acquire part of our mission. So we have 8 families, so one disappears from this phylogeny. We have 15 of the 17 genera shown on this phylogeny. And we have 62 species across the genera, 916 strains that are from 6 continents, don't have anything from Antarctica, and 49 countries. So we have genetic diversity as well as geographic diversity in our collection. So we're talking about how these fungi can be locally adapted, and that can be important. Where they come from can be really important, as well as the identity of the species. And then propagating and maintaining. So this is different from a lot of collections in that it's a living collection. So these fungi have to be grown every year to be kept alive. So we grow all 916 strains every year. We start about 20 a week and we harvest about 20 a week. And this is when we harvest them, it looks like this. We dry them down completely, pull them out of the pot, remove the biomass, and we take a sample. And I'm going to talk about what we do with those samples. And so we end up having several thousand of these cultures in storage at any given time. And then characterizing these species. So we have on our website species descriptions with a lot of detailed information about each species in our collection, and some species that we've accessed but haven't remained in our collection. And so all of these photos and descriptions are by Joe Morton, who is the curator at West Virginia. And there's all kinds of really useful information for people who would like to identify spores that they have collected. So there's like pictures of structures of this germination shield. There's sizes of spores. There's hyphal structures. And pictures of the spores themselves. This is Dentiscutata erythropa. I think it's really pretty. There's images of spore development and some of the ornamentation that can be on the spore walls. And images of wall structures. So that's one way that you can differentiate species, is by looking at the structures in the spore walls. And also, if you are really new to looking at and identifying spores, just a way to identify, is it a spore, is looking for these pretty sparkly fat globules inside the spore reflecting light back at you. So if it's a round ball and it's sparkly and shiny, that's a good sign that it might be a spore. And so one of the things that we do with these samples that we collect is we extract a sample from every single culture that we harvest in these plates, and we are trying to get pictures, not just of, so Joe's pictures are beautiful and amazing, and they show all the things that you should see, but I would like to add some pictures that show researchers what they might actually see in a microscope as well, because it can be confusing when you see the perfect thing on the computer, but then in your microscope, you're like, well, that doesn't look just like that. And we're also making slides so that we have vouchers of the spores as they were when they were accessed here at KU from West Virginia. So some of these cultures are pretty new, but some of them had been growing at West Virginia for 30 years, and the soil is different here. The growing conditions are different here. So then talking about supporting researchers as well as those species descriptions can be really useful and helpful on our website. We also have lots of methods, so you can click on these and get more lists of different methods. So spore extraction, spore cleaning, DNA analysis, culturing methods, like Liz was talking about, the little sandwich method of inoculation. And this is from the Google Analytics for our website. So we have users of our website from all over the world. And even though we're a little US heavy, we've got blue almost everywhere on that map. And then this is also from Google Analytics. This is showing views and users for the last year. And we have a lot of users, and you can see that species descriptions is one of our most popular pages, but also the third most popular link here is Kansas Biological Survey. So we do also direct traffic to KBS and the field station. And then in addition to people getting on our website and reading about our methods and looking at species descriptions, we get a lot of emails. So, and I try to answer all of the emails, and we have email conversations with, again, people all over the world and all over the United States. And these can be, the bulk of them are from faculty and students at universities, but we also get inquiries from businesses, from government entities or nonprofits. Like we've had, I've talked with the Lawrence Livermore National Laboratory and someone at the San Diego Zoo, talked to citizen scientists like a master gardener in Texas who wanted to do a restoration. And was asking about which AM fungi species he should use. So, and even a few high school students who are working on projects for their science fairs. So it's very cool that we get so much interest from so many different people. And then also we've had visitors, collaborators from Brazil, the Czech Republic, Olive from Vermont. So that has been very cool. We had a student come learn from us from Australia last summer. So, and okay, well, I don't really know what to do about that. It's not.
Defer.
8 more times, no problem.
Oh, there we go. Okay. Thank you. Sorry.
Okay. So we also distribute these spores to people all over the world, and we don't distribute them for free. We sell them. So we are funded on an NSF grant, but they would like us to try to become self-sustaining. So we have a service center where we sell cultures as well as services. So if you go to the ordering section of our website and push the store button, you will find our online store. And if you push the shop now button, you will find that we have a tiered pricing system so that it's cheaper for academic and nonprofit research to purchase our services and our cultures. And this is what the page would look like if you clicked that. So you can go in and find what we have available of each species in our collection at any given time. And that's the other reason we do these samples and extractions, is so that we can make sure there's no contamination in our cultures and provide spore counts to our customers if they ask for them. These are a little different than the commercial inocula that Liz was talking about. So a lot of those come in stinky goo or weird powder. And this is just our sterile sand, soil, and turf mix with the roots and the hyphae and the spores in there. And so also some of those commercial inoculants will say like, we have 40,000 spores per milliliter, and these are not going to have 40,000 spores per milliliter, but at least they're going to be alive. So that's something. So we've had that online store up since August, and we've had over 1,200 visitors and made quite a few sales. So that seems to be working out really well. And I guess I've showed you a lot of maps of the world and maps of the US and how big INVAM is and how broad it is, but we're right here in Lawrence, Kansas. So I wanted to finish by sort of bringing it home a little bit. So we have participated, Liz has been a guest lecturer in Ben's mycology class for several years, teaching about fungi. So we have a presence in KU. Other than just being INVAM, we participated in developing section about soil microbes for Kansas high school teachers at the Summer Institute. And we participated in developing a soil microbes lesson for the local third graders and the Kansas Ecosystems for Elementary Students program. So we want to be a part of the local communities as well as being an international presence. And I'm ending with a bunch of really cute spore pictures. Hopefully this has been a good introduction to how valuable a resource INVAM